Hello, everyone, and welcome to this second seminar, or second seminar of this VEGA seminar series. I am Simon Rue from JGI, and on behalf of the VEGA organizing committee, I'm very, very happy to welcome all of you to this seminar about phages and archaeoviruses diversity and ecogenomics today. A few quick words about the whole seminar series. Most of you are probably aware that this seminar series will have 6 seminars. We are in the second one, so there are still 4 to come on the next 4 Thursdays. And the format we try to have is in 2 parts. Right now we have the webinar part, so we will have live presentations, 2 live presentations. In between we'll have 7 flash talks. And then afterwards, we have a separate breakout Zoom session where, as an attendee, you will this time be able to ask questions and interact directly with speakers. During this webinar part and during the talk, we encourage you to ask questions using the Q&A feature in Zoom. And let me see, I think that's all the information I needed you to have. So I will hand it over to Dr. Joanne Emerson, the seminar chair.
Thank you, Simon. It is my distinct pleasure to introduce our first speaker for today, who, of course, as everyone knows, needs no introduction. So Dr. Simon Rue is a research scientist at the US Department of Energy Joint Genome Institute, or the JGI. He received his PhD in bioinformatics, ecology, and evolutionary biology at the Université Blaise Pascal, UBP in France. He is a recent recipient of the prestigious $2.5 million DOE Early Career Award for his now ongoing work to study virus-driven alterations of microbial metabolism in model soil ecosystems. Of course, as we all know, he has a long and rich history of publishing in the broad area of viral ecogenomics. It would take the entire seminar to rattle off his publication record, but I'll highlight that he's done some seminal work in marine viral ecogenomics. Of course, he's developed the VirSorter software tool for mining viral signatures from microbial genomes and metagenomes, recent work on cryptic enoviruses, giant viruses and their virophages. And he gives back to the community by organizing seminars like this, by conducting bioinformatics workshops to teach the community. And he recently led a paper, a huge community consensus paper on best practices for describing genome sequences from uncultivated virus genomes. So without further ado, please take it away, Simon. You should be able to share your slides now.
Thank you very much. Let's see. Let me check the right slide this time, put my presenter hat, and we should be good to go. Yeah, thanks a lot, Joanne, for the very, very kind introduction. So like I said, as a seminar organizer, today we are trying to talk about phages and archaeoviruses diversity as explored through metagenomics. And so this talk here will be in kind of 2 parts. One more general about these approaches and the challenges and limitations, but also the power of these methods. And then I have a second part, which is more specifically one biology story about what we learned recently about phage encoded Dgrs via metagenomics. I don't think I really need to remind this audience, but I like to start with the fact that really life on our planet is microbial. It's in the environment, in any one of us. And that means viruses of microbes are really the most important viruses ever. I have a few kind of facts of why viruses of microbes are so important. Of course, these viruses are very, very abundant in the environment, as most of you know. Viral diversity in the environment is extremely large and is mostly composed of viruses of microbes. And then you have this different impact that viruses on viral infections can have on microbial communities functioning. Of course, you have viruses killing some microbial hosts, and by killing only specific hosts, that will lead to big changes, usually in microbial community diversity and structure. Viruses will also be the driver of sometimes large-scale horizontal gene transfer events impacting long-ranging evolutionary history of microbes. And then maybe a bit more subtly, viruses during the infection itself, during their active phase, when they're replicating, they can really reprogram and completely redirect host metabolism. And I typically, especially for broad audience, try to highlight this first part, the selective killing, because this is one aspect where we think we can eventually use viruses in general and phages in particular, meaning viruses of bacteria, to really manipulate microbial community and use these phages as biotechnological tool, including for getting rid of pathogens in the case of phage therapy. That's all to say, we should want to know more about viruses of microbes. Now, how do we do this? It's really not that easy, or at least it has been a big challenge for a long time. First off, we don't have a universal viral marker gene, so we don't have 16S or 18S from which we can do amplicons and kind of survey the diversity in this kind of barcode approach. And then, of course, as again, most of you know, some by experience, microbes and especially environmental microbes are very challenging to cultivate in the lab, which means the viruses infecting these microbes are kind of one more level of difficulty. So at the end of the day, really what we need is a cultivation-free approach, which doesn't require any prior knowledge about like which viruses we need. We really just need an approach that just go and find any virus that's in your sample. And that really is metagenomics. And especially now, that's assembling viral genomes from metagenomes. So very briefly, for anyone not really familiar with metagenomics, that's a very streamlined and idealized pipeline where you take a sample, extract DNA or RNA, you do shotgun sequencing, and that gives you short reads usually, and you can then put them together, put these reads together to get genomes. You have a different flavor of this or a slight variant, which is Viromics or viral metagenomes. It's pretty much the same thing, except prior to DNA and RNA extraction, you can add steps to try to get rid of cells and only enrich for or only retain viral particles, so that most of your sequencing eventually is about viruses and about viral genomes. Now, in both cases, once you have your data, really the first key part is always identifying viral sequences in your assemblies, and then you can get a first census of what did you find in your sample, which viruses, where, which type, et cetera, et cetera. So as John already mentioned, actually, a few years ago now, almost 2 years ago now, I guess, we did publish a community-driven set of standards for performing this kind of work. So basically, this paper here, for anyone new to the field of viral metagenomics or metagenomics in general and interested into viruses, it's a very good primer because it will give you step by step, okay, this is what you should be careful with, this is what we know how to do, and this is what no one knows how to do. So again, standard tools and workflow, main biases to be aware of, and the major remaining challenges. So I really recommend this kind of community paper. Okay, so I told you that's great. I just told you metagenomics is like the best thing ever and you can explore viruses like never before. We can put some numbers of this. And this is one way I like to illustrate it. This curve here in blue is total number of virus genomes in NCBI. So, any genome of a virus. And then this in yellow is the total number of virus genomes we obtained from metagenomes. And you can see that until 2016, it was not really here. But since we have really been making big steps forward, and nowadays we routinely assemble hundreds of thousands, if not millions of virus genomes from metagenomes. Now, one way we can share this data, and one way we do actually share this data, is through this JGI-driven database called IMGVR. Actually, some of these bumps on this yellow curve correspond to the second and third version of IMGVR. If you are curious, we have a paper describing the whole database and the way we identify and analyze these sequences, and you can also access all these sequences online at this URL here. But the bottom line is there are tons of virus sequences out there assembled from metagenomes, and we can get millions of them right now. Now, of course, that seems like a very, very big number. And as you already guessed, or again, know from experience, not all genomes are created equals, right? If you do metagenome assemblies, you will also, you will very, very often get relatively short fragments assembled. So you will have a small piece of your genome and that's it. So to better kind of distinguish within these 2.3 millions, which sequences are really interesting and which ones are just short fragments, we need to get a sense for the completeness. You know, if I give you a config, can a tool tell me, okay, this represents most of your genome or only a short part? Fortunately, Stephen Neyfach has done this great work of creating such a tool. It's called CheckV. It was published a few months ago. And basically at this point, even though it wasn't here in 2019, and we only hinted that such a tool, you know, would be great, now this tool exists, and I would recommend it for, you know, post-processing any result of a viral sequence detection in a metagenome. So, you know, the bulk of CheckV will pretty much try to identify putative host regions within the contigs you provide. You should only provide a viral contig. So CheckV will try to tell you, okay, you told me this is a virus, but this part seems like it's not a virus. It will try to predict complete genomes, which sometimes assemble as quote unquote circular contigs. And by circular, I mean both ends of the contigs are identical and direct repeats. And then it will try to estimate completeness of your contig based on a database of reference genomes, which include both isolates and in a very useful way, these circular genomes assembled from metagenomes. So we cover more than the isolate diversity. We cover a lot, actually a lot more by going to these circular contigs, creating them, and then using them as reference of complete genomes, even though we don't have this in culture. So that's really, really useful. And just to illustrate how useful this is, here is the results of such an analysis on this IMGVR v3, so these 2.3 million sequences. If you take the whole database, and again, this will not surprise anyone who has done metagenomics before, more than 3/4 of the sequences are predicted to be less than half of the complete genome. So, you know, quote unquote short fragments. Then you have a, you know, 12% that are more than half. And this slice here is really the one that we are very interested in. This almost 10% of quote unquote high quality genome, meaning more than 90% complete. So, you know, roughly complete on your complete. Now, 10% doesn't feel great, but remember it's from 2.3 million sequences. So we can do the same curve as what I showed earlier, but this time only for this high quality, and that's what we have here. It's the y-axis has really changed because this time the blue and yellow curve are non-redundant high quality reference sequences. The blue being RefSeq and the yellow being this high quality IMGVR VOTUs, so non-redundant. And you can see that again, metagenome starts to be a great way of exploring viral diversity, even if you only want to look at very high quality genome. So say otherwise, you're not restricted to short fragments anymore. Okay, here is the really short success story that I like to use to illustrate how metagenomics can be a part of a much larger toolkit of viral ecogenomics. Again, I think this is a story that is familiar to a number of people here. Phage, the most abundant and widespread phage in human gut, was originally assembled from a metagenome. And that's this paper in 2014, where Bas Dutil and collaborators used 12 human gut viral metagenomes to assemble this genome on the right. This is a circular genome. What really was striking was how prevalent and abundant this phage was. And that's what's described here. The x-axis is a number of metagenome. Each viral genome was detected in. Each dot here is a genome. The y-axis is a proxy for abundance. So basically abundance on the y-axis, prevalence on the x-axis. All the brown dots are reference viral genomes available at the time. The blue dot is this crAS-phage, so clearly kind of outstanding in terms of both prevalence and abundance. Now that was both very interesting in itself, but also kind of only the start of a number of follow-up studies. One led by the next speaker, Natalia Utan, showed that this crAS-phage was only one representative of a much larger family of very abundant viruses, which again had gone completely unnoticed until we start to work with metagenomics. And then Colin Hill and his lab went through the process of using this original sequence to do targeted isolation. And eventually they were able to isolate first one and then a second crAS-phage, this second one, paper on the right, just got published a few days ago. So again, from metagenomes all the way to isolates, thanks to the original sequence. What was again made this, or at least what in my mind made this a great success story, was how well bioinformatics prediction from the original sequence matched the information we got from the isolates. We got two main predictions from the 2014 paper. One is that the morphology of crAS-Phage is likely a Podoviridae. And that was the case for the isolates. You can see the picture on the bottom left here. And then the second prediction was that the host of crAS-Phage was a Bacteroides. And that was against the case for both isolates of this crAS family so far. Now, again, that's a success story. It won't work this way every time. I think it's great to illustrate. I do want to make the point, though, that host prediction, so taking a virus sequence assembled from metagenome and guessing or predicting which bacterial or archaea or eukaryotic host it infects, is still very much a challenge, and in my mind, the major challenge faced by metagenome-based viral ecogenomics. And you can see why here. So again, this is using IMGVR-v3, these 2.3 million sequences as an illustration. And again, breaking it down by quality, you can see that even for high quality genomes, and we tried pretty hard to, we really kind of threw this kitchen thing at this. We tried every possible way to get host prediction, and we only could get good host prediction for about a third of good quality sequence. And for genome fragments, it's much lower. So that means, we still have way more than half of the sequences for which we have no clue which was the infect. Okay, so that's kind of the end of my first kind of general part. Metagenomics is a great framework for virus discovery. We have a number of standard tools and methods to do this kind of virus exploration through metagenomes. And again, I would point you to this MyUVIG paper as a great resource, especially while it's not completely out of date yet. Major challenges, I would say host linkage, really this kind of in silico host prediction is still very difficult and has a lot of margin of improvement. At the same time, there is a lot of discovery to be made. And of course, you can find completely new viruses, but you can also kind of dig a little deeper or kind of dig down and zoom in. We will find by exploring this uncultivated viruses, new mechanism for host cell reprogramming, new defense and counter defense, and so on and so forth. Basically, this is such a black box, and there are so many unknowns here that there are really, really, really a lot of discovery to be made. Okay, so that's the end of my first part. Now, switching gears a little, I wanted to use the next 10 minutes-ish to tell you about our last study of Dgrs, meaning targeted evolution in phages through the lens of metagenomics. Now, to kind of explain this story, I first need to tell you what are Dgrs. So this stands for diversity generating retroelement, and these are basically molecular mechanisms that are used to introduce mutation at very specific loci. This is explained on the right here. So you have a reverse transcriptase in red, a target gene in blue, and then you have TR and VR. These are imperfect repeats, template repeats for TR, variable repeats for VR. Now the way DGR work is this TR will get transcribed, and then the reverse transcriptase will generate the complementary DNA fragment to this RNA. But while doing so, it will introduce, or it will make mistakes or introduce random nucleotides opposing adenine residues. So that means mutations at very specific positions in a sequence that is almost identical to the VR in the target gene. And as you can already imagine, this kind of variant can then be integrated and replace the original VR sequence. So again, we typically think of mutations are kind of random and going everywhere. This is a mechanism to get mutations only at specific position within a region of a gene. So not even having mutation on a full gene, only a specific region within a gene. So Dgrs have been known for almost a little more than 15 years, actually, I think. They have been described in phages and in bacteria. And at this point, they have been also detected from metagenomes, about 1,500 from different metagenomes, including 92 recently in prophages in Forest Rural group. So really, when we started this whole project, these were the questions, right? Which phages encode and use Dgrs, and for what purpose? So these are typically the kind of questions that you can use metagenomics to address. I will kind of go through a lot of technical details and kind of bypass all this, but just so you know, blah, 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 big numbers, we mined tens of thousands of genomes and metagenomes. Bottom line is we ended up with a collection of more than 30,000 DGRs. And the vast majority was coming from metagenomes. We found them on cellular genomes and phages, many different environments. Now we took this 30,000 and we use the reverse transcriptase phylogeny to break them down into 6 major clades. And I'm not showing the tree here, I'm just showing you the different clades here, 1 to 6, and the different information, because these clades turn out to be pretty consistent on a number of ways. First, and that's this first panel on the left, DGR clades tend to be either phage or cellular. You see like phage in yellow, cellular in blue. You have 3 major phage clade, 1, 4, and 6 in bold. The 3 is kind of the only hybrid one. 2 and 5 are clearly mostly cellular encoded. So clearly phage encoded DGRs and cellular encoded DGRs have diverged a long time ago. Next, they are also found in different biomes. You can see 1, 4, and 6 again are mostly red, this kind of host-associated biome, think human gut basically, while 2 and 5 are the vast majority aquatic or sediments actually. So again, phage and cellular DGR in very different places. And then finally on this third panel, you can see they are also associated with different taxa. And here for phage encoded DGR, this is a taxon of the host or predicted host. And clearly we have a lot of firmicutes, also Bacteroidetes for phage encoded DGR, which we don't see for cellular encoded DGRs, which are in completely different taxa. So, at the end of the day, really phage encoded DGRs and cellular encoded DGRs don't share much. They only share this, fourth panel, which is this systematic adenine mutation bias, meaning apparently they have the same molecular mechanism, but a very different eco-evolutionary trajectory, let's say. That's already tell you that phage encoded DGR are something probably unique to phages. Now, what can they be used for? To try to understand this, we turn towards the target of this DGR. So again, the clades were based on the reverse transcriptase, a common part of all DGRs. Next, we look at which phages are targeted, which genes will get mutations. And kind of strikingly, we started from a collection of more than 6,000 high quality DGR target in phage encoded DGRs. And by sequence clustering, we could boil this down to only 9 protein clusters. And even more strikingly, of these 9 protein clusters, which you have at the bottom here, the vast majority could be annotated as tail fiber or protein, at least involved in the capsid decoration or tail decoration. So that's really coming from like a lot of elements, a lot of different sequences, and everything pointing towards this specific function, this specific type of protein, and these few families of proteins in phages, which are most likely involved in some kind of host attachment or recognition. So that was pretty interesting. And that kind of led us to think that most likely there was some kind of host interaction going on here. So that's where, again, mentioning Stephen, Stephen led a very interesting analysis trying to understand if DGR targeting tail fibers could maybe unlock more hosts for these phages. And it turned out that it may be true. So the way Stephen did this was taking from the same metagenomes, phages that encode DGR and phages that don't, and then matching these genomes to CRISPR spacers, so a vast reference of host CRISPR spacer, and trying to understand if the diversity of CRISPR spacer linked to DGR encoding phages, DGR plus in this case, was larger than the DGR minus, the one without DGR. And that's what you have at the bottom right. Clearly, you have a larger diversity of host species normalized by protospacer and length, et cetera, et cetera. But basically, after all normalization, you have more host species connected by CRISPR to the DGR plus than DGR minus. No, that doesn't necessarily mean host range. It most likely mean host access. Again, a CRISPR spacer match mostly means that you got an unsuccessful infection, right? The phage came in, but got like destroyed and CRISPR spacer was acquired. So it doesn't mean that the DGR positive or the DGR encoding phages can really infect more hosts, but they can probably access and inject their DNA into a larger, a broader range of hosts, which may be very useful in some biomes. Okay, last thing that I wanted to share, and because that's another aspect that I think is really interesting in metagenomes, is in the case of DGR, we were wondering, okay, that's great, that looks really interesting, host interaction, but when are DGR active? And my intuition was like, DGR, it's introduced mutation, it may be risky, so it's maybe super tightly regulated and only active during stress. No, by doing read mapping, so looking at individual read variants instead of the consensus sequence that the metagenome assembly gives you, we can actually get a sense for it. And that looks something like this. So what's represented here is one DGR target at the amino acid level. So you have the consensus amino acid here, which will be the translation of your predicted protein from your contig, but then you can look at individual reads and you can ask the question, the read mapping to this region, does this match exactly the consensus, or is there some variation there in terms of the amino acid encoded? On the left, you can see, we are piling up individual reads in rows, but there is very little variation. That's what we would call no sign of active DGR. On the right, it's the same consensus sequence, but you see many, many more variants, and reads are very different. So that's what I would call high variability and active DGRs. And we kind of put some stats behind it, but that's the gist of it. No, by doing this, we can classify, all the DGR and ask whether there is some sign of recent activity. And that's what we found. And again, these are my 6 clades of DGR. You have the 3 phage in bold. More than half, and actually more than 2/3 of the DGR show some kind of recent activity. So that means that my idea that it was all very tightly regulated and stress-bound may be wrong. And we actually validated this. I don't have time to show this, but we looked at time series and we could show that between time points, we not only see high variability, but we see a high turnover between alleles and between amino acids. So basically, we seem to see a pretty constant and recurring activity of DGRs, which kind of had me flipped and say, okay, it may not be so tightly regulated, but it may be used to maintain a very high population diversity at target loci. So almost as a proactive way, making sure that whatever happens, the population of phages in this environment always have a pretty broad diversity of tail fibers, et cetera, just in case, basically. Okay, conclusion. Again, using metagenomics assemblies mostly, we found a lot of different phage DGR loci. We were able to link them to a potential role by doing some annotation and kind of follow-up studies. Most targets are likely tail fibers involved in host attachments. Most DGR seems constantly or very frequently active. And to me, this story also illustrates the strengths of metagenome exploration, meaning we have a very large uncultivated phage diversity that we can now search because we have these high quality genomes and we have this database, and we can then ask specific questions on a molecular mechanism or a specific gene family, these kind of things. And really then it's really primes you to do follow-up targeted experiments in the lab. And with that, I'm probably out of time already. So just want to thank everyone, and at JGI, collaborators on DGR study in the IMG team for the IMG VR. And I highlight in bold Stephen Neyfach here because I mentioned his name multiple times and you have seen how I kind of relied on most of his work. Thank you.
Wonderful. That was a fantastic talk, Simon. Thank you so much. So yeah, there's time for a couple of questions. So I'll check the Q&A and please feel free to start typing those. So there is one question so far. Is there information on genes that bacterial DGRs tend to target similar to phages targeting tail fibers?
It's much more difficult. I kind of glossed over the details, but when I say it's phage tail fiber, I'm kind of embellishing a bit. It's phage tail fiber if you believe and if you trust the kind of very new prediction of phage protein structure, like it used to be called ivarian. No, it's called FANS, I think. It's from Anka Segal. But basically, we don't have sequence similarity. We only have like amino acid composition suggestion that is a tail fiber. It all makes sense though. For the cells, it's a bit of the same story. We don't have sequence similarity. It's a lot of unknown genes. What we seem to have is a lot of transmembrane domains. So it seems to be membrane anchored proteins or secreted proteins, but that's as far as I can get.
Great. All right. So the next one, thanks for a very interesting talk, Simon. I was wondering how the prevalence of phage encoded Dgrs varies in different ecosystems.
Yeah, it does vary a lot. We see, even when trying to normalize for like diversity, we seem to see a lot more phage encoded Dgrs in, let's call them tight environments, especially human gut, rumen, other mammalian guts actually, and much less in kind of low density environment. We see more cellular encoded Dgrs and many less viral encoded Dgrs in freshwater and marine environments and these kind of things. I have this theory that this is linked to difference in host density and the way host defense works, and the way that in some cases, and under some selection pressure, hosts will tend to defend themselves by modifying their cell surface, while in other host density and dynamics, they will tend to defend themselves by CRISPR, for instance. And Dgr is useful in the first case, not useful in the second one. So I'm guessing there is something at play here. I don't have the full picture yet figured out, but hopefully someone will.
Great. All right. I think we have time for one more, and this one is coming from the chat. So only the panelists will be able to see it because this person couldn't type it in the Q&A. But the question is, I'm curious how these tools like CheckV recommend handling bins of phage genomes.
That's a good question. I think CheckV doesn't handle bin natively. So you would have to stitch together the contigs from your bin and present it as a single contig if you would like to have a completeness estimation on the whole bin. It will work though, because CheckV is all based on predicted genes. So once you stitch together your contig, CheckV will predict genes all across. Most likely it will have the same gene prediction as if the contigs were separated, and that's fine. It's a bit more difficult for larger viruses for which genome binning is required. I would argue binning phages right now is still useful, but will be less so in the years to come, just because sequencing depth and everything means that we'll have phage genomes in a single contig. For larger genomes, like giant viruses, there you need genome binning, and there we have not yet a good way of looking at completeness.
Excellent. I think there are lots and lots more questions. I think in the interest of time, we're going to move on, but Simon may be able to type answers to some of these as we go on. So yes, thanks again for a fantastic talk, Simon.
Thank you very much. Okay. Now again, switching pace a little. This was a long talk. Here is some quick, fast, and really kind of breezy flash talks. And I think Manu is the first one. Go ahead.
Okay, I'm gonna start my talk with a question. Does viral quasi-species exist like RNA viruses? But first we need to explain what is a viral quasi-species. That basically is a viral cluster with a lot of number of genetic variants. It's important microdiversity because it determines, for example, pathogenicity. So we discovered a few years ago in my group, one of the most, or probably the most abundant virus in marine system that was infecting Pelagibacter was discovered by single virus genomics, and it's called 37F6, that virus. Now we know that it's not only the most abundant, but probably is one of the most microdiverse virus, because we discovered up to more than 1,000 viral strains in a single sample, and more than dozen species in just one sample. So the answer is probably yes, we do have DNA viral quasi-species probably in nature. So thank you very much for organizing this.
Yeah, I'm not even sure I want to think about this. Thank you.
Microdiversity matters. So I'm not sure if you read what it's in my heart. Thank you very much.
Thank you. Yeah, I'm not sure I can even wrap my head around this. So let's move on. Miriam.
So hello, I'm Miriam. I'm a PhD student in the Sunagawa lab at ETH Zurich in Switzerland, and I am interested in temperate phages, which are recognized as modulator of the gut microbiome. However, challenges in identifying and locating inducible prophage in the host genome, as well as quantifying their activity, have limited our understanding on like prophage-host interaction. And this motivated us to like develop and experimentally validate an analytical framework for sequencing data to identify and precisely locate inducible prophages in the host genome. And for that, we are focusing on like paired rates that are flanking or covering the attachment site of induced phages. We identify phages by viral reads, which reveal reverse forward orientation, and we align them back to the reference genomes and precisely locate them by viral reads, which are only partially aligned with reference genomes. With this analysis method, we identify and precisely locate inducible prophages and subsequently determine this, their phage activity, as you can see here as an example for Akkermann's Cinephilia. When we then compared our predicted location to the results of prediction tools, we could see that the tested prediction tool could not or had problems to precisely locate them precisely and non-ambiguously, and the results were different between the prediction tools. When we then further applied our methods to more complex microbiota community, we could see that we were able to detect induced prophages even in strains which had less than 1% relative abundance within complex communities. So if you're interested, you're really welcome to talk to me in the breakout room, or you have the opportunity to read the paper which we recently published. And the MBS tool to like detect and locate these inducible prophages is freely available on GitHub. Thank you very much.
Thank you. That's great. Okay, Gary.
Hi, everyone. I'm Gary Trubel. I'm a postdoc at Lawrence Livermore National Lab. In any given environment, microbes can exist in many states, including deceased, dormant, or active. And I'm showing you two examples of how we can use stable isotope probing combined with metagenomics to look at certain organisms. So in the first example, we use heavy water, which is H2-18O, and we can look at all active microbes and their viruses in an environment. We incubated Arctic soils under sub-freezing and anoxic conditions for a full year, and we discovered that 1/3 of bacteria were active, whereas 2/3 of viruses were active. And we realized by the end of the study that we would have overlooked this community had we just used a traditional metagenome. In our second example, we have tropical rainforest soils that we incubated for 44 days with 13C enriched plant biomass to look at who's breaking down this plant biomass. We discovered, comparing traditional metagenomes with CYP metagenomes, that we were able to recover more viral populations from our CYP metagenomes, that oxygen content really drove the viral community, and that the active organisms were infected by active viruses, and that they were key to breaking down carbon in this environment, showing that viruses may be implicated in controlling the fate of this carbon. So this is just an example of how we can use stable isotope probing to look at all community members or specific ones. If you want to know more, talk to me in the session. Thank you.
Really cool. Thanks. Elena.
Hi, everyone. I'm Elena, a PhD student in Frank Aylward's lab at Virginia Tech. And I work with jumbo phages, which are phages that have genomes over 200,000 base pairs in length. They often have large particle sizes and can't fit through filters that we commonly use to enrich for phages, which is that 0.22 micron. And so this led us to ask, how much are we missing them by using these filters? And we used this Terra Oceans data set. Oh, shoot, I'm losing things. Okay. We used these Terra Ocean metagenomes to ask this question because they've gone around the world collecting seawater samples and using different filter sizes. And Terra Oceans has also collected metatranscriptomes at many of these sites. So we could also ask how active these phages are and where they're found in the ocean. And then we were also curious about how these jumbo phages are related to phages of other sizes. And so we did some analyses against all the complete genomes on RefSeq. And we found that the 0.22 micron filter reduced our ability to recover jumbos. About 20% of the jumbos we were working with couldn't be found in samples below 0.22 micron. And then when we looked at the distribution based on read mapping and activity based on transcriptome read mapping, we found that they were most abundant and active in coastal biomes. We think this might be because nutrient concentrations are higher there and potential host cells may be bigger there. So it's more likely that a jumbo can infect something where there's bigger cells. And then we also found that these jumbos are more related to groups of phages of smaller sizes, which suggests potentially that they have independent origins, which has been found in some other studies. But if you have more questions, I'd love to talk more about jumbos. And yeah, let me know. Thank you.
Jumbo phages are really cool. Thank you. Marina.
Hi, everyone. I'm Marina, a PhD student from the University of Alicante, and I'm going to talk to you about deep sea phasing of RNA viruses. Despite the abundance and importance of viruses in aquatic environments, we still have very little knowledge about their distribution and the composition of their populations, especially for RNA viruses. To address this matter, we have analyzed different water samples from the Atlantic Ocean down to 4,000 meters to search for RNA viruses using hidden Markov models for the RNA-dependent RNA polymerase. Thanks to that, we have discovered a lot of new RNA viral sequences yet to be classified and some interesting data about RNA viral populations, such as the stability of their composition between different samples. So if you want to learn more about this, please check my poster and feel free to ask me any questions. Thanks.
Thank you very much. I would not have guessed RNA viruses at 4 kilometers deep, I will admit. Francisco.
Hi, everyone. I'm Fran from the University of Alicante, Spain. In recent years, our knowledge of the diversity of the marine biosphere has been highly expanded, thank you mainly to Viromics. On the other hand, single virus genomic has appeared as a complementary method that allows us to recover the genome of highly microdiverse viruses, which assembly from Viromic source read is hindered. Here, we have implemented single virus genomics to the dark, deep ocean, using samples from the oxygen minimum zone and bathypelagic regions obtained from different oceans and the Mediterranean Sea. This methodology has allowed us to expand the known diversity of the deep ocean using very small seawater volumes, only from 1 to 10 milliliters. Thank you for all the attendance.
Thank you. That's really neat. And last but not least, Alessandro.
Thank you. I'm Alessandro Rossi. I'm a PhD student at the University of Padua. Now, in anaerobic digestion, the combined activity of bacteria and archaea leads to the production of biogas. The viral community, though, isn't very well known. For this reason, I devised an experiment consisting of several different batches, each one of them undergoing a different stress in order to induce proviruses. Viral and microbial components were separated by filtration and sequenced, and then the ensuing bioinformatic pipeline yielded more than 1,000 viral genomes. Now, the redistribution shows no substantial difference across the conditions. The viral genomes, though, are mostly single contigs and bin genomes, and only a fraction of them is predicted as integrated. Overall, these recoveries paved the way for more analysis, aimed at understanding the role that the viruses play in this microbial community. Thank you very much.
Perfect, thank you. And thanks a lot to all the Flash Talk presenters. Again, I wish we could do round of applause, but of course it's Zoom and everything, but really great work. And it's not easy to just distill everything down to 1 minute. So thanks a lot to all of you. Okay, and with that, I will hand it over to Joanne again to introduce the second talk of this seminar.
Great, yes, and I just want to echo Simon's comments. Those were fantastic Flash Talks. Always a pleasure to get an update on the latest research. So it's my pleasure to introduce Dr. Natalia Utan. She's a staff scientist in the computational biology branch at the National Center for Biotechnology Information, better known as NCBI, we all know and love, at the National Institutes of Health, or the NIH in the US. She received her PhD in the Department of Biology, Technion Haifa in Israel. And she's really a pioneer in cutting edge research on the comparative evolutionary genomics of viruses from really quite a wide variety of groups. I'll highlight a few here. So for example, she's done a recent work on crAS-phages in the human gut, which we've heard a little bit about, and we're going to hear a good bit more about, I think, in her talk today. But she's also worked on NCLDV evolution. She has a cool proposal on the megataxonomy of the virus world. And she's been doing some interesting work also on viral and bacterial rhodopsins. So her background and interests really span quite a wide range in virus evolution. So without further ado, please take it away, Natalia, and you should be able to share your slides.
Thanks, Jen. I'm trying to share. Do you see my screen?
Yes, looks great.
It's great. So crAS-phages do not need much introduction after Simon's talk. They are abundant in human gut virome, and they comprise an expansive group of viruses only distantly related to other phages. crAS-like phage genomes contain 3 gene modules for replication, transcription, and assembly. Replication block contains DNA G primase, either family A or B DNA polymerase, several other replication genes specific to crAS-phage. Transcription block contains long multi-domain proteins and code for RNA polymerase subunits characteristic for crAS-phages. Assembly block contains capsid terminase portal proteins, as well as some other proteins, again specific for this phage group. Comparison of crAS-phage proteins to genomic and metagenomic sequence databases revealed several hundred phage genomes that were to different degrees related to crAS-phage. Note that human-associated phages that are red on this tree do not form a monophyletic group. They are mixed with phages from various other environments. Analysis of 250 genomes of human-associated crAS-like family members led to proposal of 4 candidate subfamilies and 10 candidate genera of crAS-like phages. When I combine these 4 genera with the tree I showed previously, I have this kind of tree. The 4 proposed subfamilies are here, and they lie pretty well within the previously known diversity of crAS-like family. To expand the diversity, we screened circular contigs of human gut metagenomes for virus-specific genes. We found 596 contigs belong to extended crAS-like phage family. I said extended because from this screening, we got 2 groups that I named just following the lead of the previous study. I named epsilon and zeta. These groups differ by the genome size from the original crAS-phage. Here, the zeta group is almost twice longer as the original one, but the monophyly is supported by terminase tree, by other protein trees, and by the set of genes that are characteristic for crAS-like family. Despite significant difference in genome length, these 2 new crAS-like groups share the same genome organization. They have same 3 gene modules and the same set of conserved genes. Abundance. Among the sequence reads mapped to the putative phage genomes, 86.7% are crAS-like phages. The alpha-gamma group accounts for half of them, 53%, and most of them are phages closely related to crAS phages. What is interesting here, the two new groups, zeta and epsilon, account for quite a significant part of human gut virome. These phages were never seen before. So the question is, how come that phages that abundant were unseen in previous studies? The first answer is alternative coding. Typically, annotation of a phage genome starts with predicting its protein coding genes. Orf's the original crAS phage is a good one. It is predicted with standard bacterial code, and where the proteins are, we can see open reading frames, meaning places without not interrupted by any stop codons. Another phage of crAS-like family would look like that. We have short open reading frames with long intergenic regions and no open reading frames here. Translating the genome with a modified code with TAG reassigned to glutamine recovers all the proteins characteristic for crAS phages. Here are long translation proteins. Alternative genetic codes in phage genomes have been reported previously, especially in human-associated metagenomes. Altogether, we identified 243 crAS-like phages, mostly from beta, delta, and zeta groups that use alternative codes. One of the common mechanisms for codon reassignment employs suppressor tRNAs. Many genomes of crAS-like phages, especially in the beta and zeta groups, encode multiple tRNAs, including putative suppressors. The delta group phages have the highest frequency of suppressor tRNA. crAS-like phages apparently can switch genetic codes at short phylogenetic distance, as exemplified by this zeta group tree. For example, this branch has TGA reassigned, and this branch has TAG reassigned, and sequences in the middle have no detected stop codon reassigned. However, the evidence of codon reassignment is not in a perfect agreement with the presence of suppressor tRNA. Here, for all this branch, only one genome has suppressor tRNA corresponding to this reassigned, and vice versa case is here. The whole branch contains suppressor tRNA, but no reassignment was detected. We traced several cases of suppressor tRNA emergence. In this case, a suppressor tRNA emerged via a single mutation in the anticodon tRNA. There are 6 closely related species. Here is an alignment of the corresponding tRNA, and one point mutation produces the TTA anticodon, which makes this tRNA a suppressor tRNA. However, we would not find signs of stop codon reassignments in this particular genome, and this might be an apparent case of suppressor pre-emergence, meaning that this phage doesn't have any stop codon reassignments, but its descendants probably will do. Another case of suppressor tRNA emergence, one genome of beta group phages acquired a TAG suppressor tRNA, probably from a bacterial source. And this genome started to accumulate TAG codons in the conserved genes such as terminase L. Here is an alignment of terminase large subunit of this genome and related genomes, and there are multiple places where glutamine is encoded by TAG in this particular genome and not in others. When I translate this genome in standard code and in standard with TAG reassigned, we can see several places where this coding modification would make a difference. Without this modification, we see disrupted frames. So TAG reassignment does happen in this genome, but probably in beginning stages. Another feature of these phages is very high abundance of introns and inteins, and especially phages of zeta group, which was unseen before, are affected. They harbor numerous introns and inteins, and specifically in conserved genes, in marker genes that we use to use for screening of metagenomes for viruses. And this massive infestation with introns and inteins severely complicates our screening and our annotation of these genomes. For example, this capsid gene is split into 5 segments across 2 coding frames, and this DNA polymerase is split in 4 segments interrupted by introns, and on top of that, it has multiple stop codons. And in this example, we have a terminase gene that is encoded in 3 in-frame fragments separated by inteins, and through the whole length, we see abundant TAG stop codon. Next genomic feature of these phages that we didn't see before is the frequent switch between family A and B DNA polymerases. With the exception of the epsilon group, crAS-like phages encode either one of them, and the alpha, gamma, and delta group have family B DNA polymerase, which is purple here, and zeta and beta group has they have both. I marked sequences with genomes with family A by red, with family B in purple, and we can see some fine mixing between these 2 families. When we look at genome locations of this polymerase, we notice that they share the same genomic context, the same positions. So we began to investigate what would happen here, whether one of the polymerases was ancestral and another was horizontally transferred. For this, I made the phylogenetic trees for both polymerases, and the answer was neither one is ancestral to all the crAS phages, because no tree follows 5-group phylogeny of crAS-like family. On both trees, we could see that all crAS-like phages are grouped together, with some exceptions, with probably polymerase was taken from a bacterial host. But within crAS-like phage group, we have a good mix of beta and zeta phages here, or beta, zeta, and delta phages here. So we never see before, we didn't expect this kind of thing, because Polymerase gene meant to be a core gene that we use to trace phylogeny, to establish taxonomic relationships, and it was not known as a gene to be prone to horizontal gene transfer or any other kind of jumping. When we look at what else travels with polymerase, we looked at several examples where we have like a couple of closely related phages that are similar to each other, but the polymerase, and this polymerase is similar to polymerase from another group. And when I look at nucleotide alignment of flanking genes and polymerases, I see the surgical precision of the insertion, the switch between family A and family B polymerase, and answering nothing travels with the polymerases, and the mechanism of this gene replacement is unknown. When I try to reconstruct the history of the polymerase switch, I would get a picture like this. I took the same zeta group tree that I showed before and mapped probable transfers from A to B and polymerase zeta group to beta group. So we can see that they do the switches on many occasions quite casually, and is fascinating because we never see this before, not in phages, not in other genomes, and probably we think that it might represent a new distinct defense mechanism characteristic to bacteroidetes. The last feature that I want to show is variable gene boundaries. Again, the zeta group is an example. This is a transcription region of zeta phages. All of them are close to each other to that extent that I can align them, align the genomes. They all have domains characteristics for crAS-like phages, RNA polymerase A, B, and B prime. But the gene boundaries look quite arbitrary. Like in this phage, we have all the domains encoded by one protein, two proteins here, and so on. And alternative coding and the presence of introns and inteins do not account for this seemingly arbitrary gene boundaries. These features possibly pose an extra challenge for genome annotation and analysis. And again, probably they represent another yet unknown mechanism of gene translation or post-translation, post-transcriptional and post-translation modifications. As of a summary, we analyzed around 5,000 circular metagenomes. We found nearly 600 genomes of crAS-like phages. They account for nearly 87% of the DNAs mapped to this circular context. Out of phages that we found, two groups were unseen previously. They are completely new. Originally discovered crAS phage was an outlier in terms of genome organization. It has no introns or alternative coding schemes. which means that we were lucky that this came first, and we were able to analyze and annotate it. And this new recently found family members are not so easy to annotate. Some of the crAS-like phages have genomic features that we didn't see before, like abundance of introns and inteins, and alternative coding, and frequent switch on DNA polymerases. That's it. I want to thank members of our group that participated in this study, and our collaborators that helped us with metagenomic data mining and preliminary analysis. And thank you for your attention.
Wonderful. Thank you for a fascinating and very rigorously researched talk, Natalia. So we'll take some questions. So there's one right now, which is, do you know if genomes with different reassigned stop codons coexist in a single population? That is the switch happening consistently within a single population, or does this segregate by environment?
Oh yes, all these phages came from the same environment, from human gut. If you did I answer?
Yeah, and then so I don't want to infer what the questioner might mean, but I think is it within a single population? Are there are there these reassignments basically probably happening consistently within a single population, or are there a whole bunch that are just consistently existing in the same community? So clearly they're all existing in the community. What about the population?
We don't have a population. These phages are from metagenomes, so all we have are metagenomic assemblies.
Yeah.
Nobody grew them in culture, so we can't operate populations. It would be good to know, like it would be interesting to see in person how fast, how often it goes on. But so far, none of them is in culture.
Very cool. Okay, so the next question, thank you for a wonderful talk. Apart from DNA polymerase, do you find other genes that show the same excision pattern? And do you think homologous recombination between two different phages infecting the same microbe does not explain it?
None. No other gene have this pattern of excision. So far, this is the only one that we found.
Great. If there are, let's see, we have one in the chat here from Simon. Can polymerase really be swapped that easily, or are there some other genes that need to co-evolve or adapt for efficient replication?
Okay, so I investigated this specific question, and I found no other gene or cassette or whatever in this location, like flanking sequences do not contain them. Maybe there are some complicated mechanisms that is located everywhere else in the genome. I can't say, because there are still lots of uncharacterized gene in every genome of these phages.
Great. So I actually have a question related to the introns. So are those exclusively bioinformatically identified where you're seeing the split, or do you also, from the genomes, or do you also, for example, have metatranscriptomic data that shows that, there's some splicing, or, yeah?
No metatranscriptomics, and even no intron prediction by bioinformatics. Because there are very few letters and they are variables. So what we did, we looked for intron, intein, encoded endonucleases to see how many introns there are. And probably there are more introns, so we are on the safe side, because I imagine that there are many introns without easily predictable endonuclease, but at least on this part that does have endonuclease inside, there are a lot. They are very, very abundant that we never saw in phage genomes before.
Yeah, it's absolutely fascinating. So I think that's it for questions at the moment. Or did I miss one? Let's see. I did miss one. Here we go. Okay. So how do you think host abundance influences the presence of these diverse coding schemes and repertoires, or in terms of competing against other phages?
I think that the human-associated communities like oral metagenome, gut metagenome are tight. They are pretty well packed with microbes and viruses, and it encourages competition, and this encourages developing such strategies. The study, the first study on this topic was Science 2014, Stop Codon Reassignments in the Wild. They did an excellent job on screening of metagenomes for possible alternative codings. And yes, human-associated metagenomes are champions in frequency of alternative codings in phages. So I would say that in marine and marine metagenomes where microbes are not that abundant, we would not expect that many tricks with alternative coding or else.
Great. Okay, well, thanks again for a wonderful talk, a really fascinating system and thorough set of analyses. And I'll hand it back over to Simon to wrap things up before we head to the breakout rooms.
Thank you. And yeah, like echoing what John just said, thanks a lot for a great talk and a great session all around. With that, I will invite you now to join the breakout session, which you have the URL here. You should have this in your original registration email as well. And also, if you would like to know more about viruses and keep kind of having this and attending these kind of talks, join us next week for a SARS-CoV-2 special seminar by Eddie Holmes. And then after that, we'll come back to the regular format for seminar 4, 5, and 6. So with that, we can close this seminar specifically. And again, thanks a lot for joining, and see you all in the breakout session. Thank you.